Kia ora everyone, so welcome to the next mini online lecture. And what I want to do in this lecture is apply the knowledge that we came up with from looking at phylogenies to a problem which is the nature of fungal pathogen outbreaks. And so what I'm going to be doing here is talking specifically now about fungal pathogens, so not about fungal mutualists, but about the pathogens, and looking to see how we can apply our knowledge of phylogenetics to understand more about the origins of where pathogen outbreaks come from. So I'm going to turn off my video and start going through the slides. Okay, so fungal pathogen outbreaks. So fungal pathogens are common, and they infect both plants and animals and all sorts of other organisms as well. We're going to largely think about this in the context of plants, although be aware that you can apply these ideas to fungal pathogens of anything, and in fact, not just to fungi, but to other pathogens. So typically we think of fungal pathogens as being an arms race with their hosts. So if we imagine that we have a plant host, and then it's existing in a big population, that population is diverse, and then there's some pathogen that's infecting that host that also exists in a population that's large, and that population is also diverse. So they've kind of coexisted alongside one another for some time, and they're kind of involved in an arms race kind of dynamic, which you might have heard of before. And so that arms race dynamic goes something like this. So let's imagine in that pathogen population, you have a more virulent mutant arise, and then that can then, because it infects more hosts, it spreads throughout its pathogen population. But then the host will evolve some sort of resistance mechanism to that, and then hosts that are resistant to that pathogen, then they'll spread throughout the host population. And so you'll get these kind of arms race dynamics going on. Now, one of the things that we find is that when this when these arms race dynamics occur, the population of the pathogen and of the host, in fact, both remain high. And this is because of the activity of sexual recombination in sexual species that's able to spread around virulence and resistance mutants around the population without lowering the diversity in those populations. Now, it's not important if you don't understand what how this works. So if the way that sexual recombination is able to maintain high diversity despite the fact that virulent and resistant mutants arise in the two populations, that's fine. If you just take my word for it that that happens, that's fine. It's not a part of this course to understand why that is. If anybody's interested, I'm happy to explain it further. But for the purposes of this course, you just need to know that that's the case. Okay, so that's the kind of basic picture of what we think happens in normal kind of populations where there's host populations and pathogens that are attacking that those host populations. But what we find is that there are fungal pathogen outbreaks. And we'll talk in the next slide about how that works. But the dynamics for these pathogen outbreaks are different. And so what we're going to use our understanding of phylogeny for is to understand whether we see some sort of virulent mutant arise, whether it's due to the process that I talked about in this slide, or whether it's due to the pathogen outbreak dynamics that I'll talk about in the next slide. So let's go to slide number 2. And now what I'm talking about is fungal pathogen outbreaks. And so there's a couple of ways that you can get outbreaks of fungal pathogens. So first is that a fungal pathogen can be introduced to a naive host in a new area. And in Aotearoa, we're familiar with these kind of dynamics because we don't have a lot of pathogens. And so, for example, the myrtle rust pathogen that some of you may have heard of has been introduced into New Zealand. It's never been seen in New Zealand before. So we have a series of naive host plants that are susceptible potentially to myrtle rust that have never seen the disease and therefore never developed resistance to it. The second way that you'll also be familiar with is cultivated crops. So usually these are planted as a monoculture, which means that they have low genetic diversity. Genetically, the plants are all extremely similar to one another. And that means that the all those individuals in those monoculture populations are equally susceptible to being infected by a fungal pathogen. If you happen to have a pathogen that is virulent against one, it'll be virulent against them all. And so in both cases, the pathogens are characterized by outbreaks of a single virulent lineage that sweeps through and has low genetic diversity. And I'm just in this diagram here, I'm showing an example of where this has happened. So this is the spread of wheat stem rust. So this rust is a fungal pathogen, a Basidiomycete mycete pathogen, and wheat rust is a very important plant pathogen economically and for food security reasons. And so there's cultivars of wheat are resistant to rust, but there's a lineage of wheat stem rust arose, it's thought, in Uganda here, and then in the ensuing 10 years has spread both north and south through these monoculture wheat crops that are all susceptible to infection by this particular rust variety. Okay, and so this difference, whether a fungal pathogen comes from just a virulent mutant arising from an indigenous arms race situation, or whether it's one of these outbreaks that have come because of the host being naive or being something like a monoculture, is really important for understanding and preventing fungal infections. And it's also important for developing resistant host varieties. So if we move to the next slide, what we're going to do is look at the predictions or different predictions that these two fungal pathogen scenarios provide. So what we want to ask here is two simple questions regarding a new fungal plant pathogen. So let's imagine we observe, we have some sort of host, and suddenly we observe a fungal pathogen appearing on that host that we've never seen before. And so we have two simple questions that we want to ask about this. The first is that outbreak caused by an indigenous pathogen variety that has become virulent? So what I talked about in that first slide. Or has it been introduced from somewhere else? Or if the plant is a monoculture, is it a pathogen that's able to infect all the individuals in that monoculture? And if it's the latter, then the question is, where has the pathogen come from? So where does it originate from? So we can learn more about it. And the key thing that I'm going to talk about is that we can generate predictions. So different predictions for these questions, depending on what the answer is. And these questions all relate to diversity of the populations. And when I talk about diversity, I mean genetic diversity. So how much genetic variation are there between individuals in the population? And we're going to be looking at this from the perspective of the fungal pathogen. So how much genetic diversity in the fungal pathogen. Okay, and so what are the predictions for genetic diversity of the fungal pathogen made under these two scenarios? And so the two scenarios are if the outbreak was caused by an indigenous pathogen variety that evolved extra virulence, or if the pathogen was introduced from elsewhere. And so I'm going to get you to think about what those predictions might be. But before I do that, I'll give you a bit of a clue and talk about the nature of genetic diversity and how genetic diversity can change. And in particular, I'm going to tell you about one phenomenon in population genetics, which is the idea of bottlenecking. So here we have a picture, and let's take the bottle on the left. So this bottle is a representation of a population, and we might imagine this to be a fungal pathogen population. And all the little marbles or balls or whatever these things are in the bottle are individuals within that population, okay? And the different color represents different genetic variants that are present in that population. Now, this picture is not particularly great because there's not a lot of colors. Ordinarily, we'd expect there to be way more colors, but anyway, just for purposes of illustration, it works fine. And let's imagine that we take some of those individuals from that bottle and transfer them into a new container. So we tip the bottle upside down and a few come out. And now if we look at the composition of those, we can see that the composition in terms of diversity, in terms of the number of different genetic varieties, is reduced compared to the host population. And so this phenomenon is called bottlenecking, and it's the bottlenecking of the population. And it means that if you take a subset of individuals from the kind of main population and introduce it into a new area or a new host, that necessarily you have the genetic diversity of this new small population will be reduced compared to the entire population. So that's the bottlenecking phenomenon. So based on that, if we think about 1 and 2, if the outbreak is caused by indigenous pathogen versus if it's a pathogen that's introduced from elsewhere, what would your expectation for the level of genetic diversity be? And I'll give you a couple of moments here. If we go to slide number 5, which is just a blank slide, I'll give you a couple of moments. I'll give you 2 minutes, in fact, to think about that. Okay, so if we go back to the slide number 4, how did we do with that? And I think if you've been following along with what I'm saying, the answer is kind of obvious. The answer is if the outbreak was caused by an indigenous pathogen variety that involved extra virulence, it would still be part of the same population. And so the genetic diversity in that population would remain high. Whereas if the pathogen was introduced from elsewhere, you'd have this bottlenecking effect, and therefore the genetic diversity would be low. And so the key prediction between these two scenarios is that in the top one, the genetic diversity will be high, and in the bottom one, the genetic diversity will be low. Okay, and if I go on to the final slide, slide number 6, I'm just going to quickly cover how we measure genetic diversity. And usually we do this by measuring DNA sequences. And the way that we do this is to PCR amplify a region of the genome. And usually we pick a region that's we think is quite highly variable. And then we characterize it somehow. And an easy way to imagine this is by sequencing that DNA. And we can look at those DNA sequences from different individuals in the population. And where we find DNA sequences from different individuals are very similar to each other, we say that those populations are clonal. And if we have, conversely, if we find that different individuals each have different DNA sequences for that particular region, we say that those populations are diverse. So a clonal population is indicative of a pathogen outbreak, whereas a diverse population is indicative of the pathogen being an indigenous pathogen where some of them have having to evolve virulence. So that's all I want to say for this mini lecture. And what we will do in the next one, the final one, is go on and apply this knowledge now to looking at some examples of fungal pathogen outbreaks. So I'll finish there. And again, if you have any questions, please direct them to Piazza.